Great. So, yeah, so in this slide that we mentioned, the term that we all have heard many times, medicine of the futures. So what is the medicine of the future in the post-genomic area? So there's a lot different definition, but the three, that would say, different characters, I think, personalized, predictive, and preemptive really characterize very well the type of medicine we want to see. So when we think of personalized, we mean that we have to have a linkage to our own genome. Okay, it had to be predictive. That means that can we detect disease before people know they are sick, and also preemptive in the sense that if we detect earlier, then of course the management of the disease is much better. So when we think about early detection, we look at this slide, for example. In general, when people they get sick, they feel a symptom, and then they go to the doctor, and the doctor will diagnose, oh, we have this, and get to treat. But now what we want to do is that, can we detect before people, feel that they or know they are sick? And that means that we need to go upstream. And in this sense, I mean that we have to go to at the gene level. And for that, that's where for the last couple of years, we have been involved in, can we detect those microRNA, mRNA biomarkers sensitively early enough to prevent or to diagnose that early? So that is the philosophy I'll go for the last couple of years. So this is a slide that all of us know, maybe for the group of session, they know, but maybe for the general public, we work on the so-called Raman and surface enhanced Raman. Very quick, 5 seconds. The idea when you have nanoparticles and you shine light to nanoparticles, it makes the condition electron oscillate and it creates a so-called strong plasmonic field. So the only thing that we need, I would say, to notice that the so-called the lining rod, then we have a very sharp curvature FIP spike. Then at the tip we have create a strong enhancement, and sometimes people call it a lining rod. So just remember, it will come back later. So when we want to detect, genomic kind of a probe, we create this kind of a probe, and we call molecular sentinel. So we designed a probe which has a hairpin loop that is, designed to detect a sequence, and when nothing happens, it forms a stem loop, and we have here a Raman label. So if we have a particle, which, for example, a particle of silver, then you have it make a stem loop, and the dye is very close to the lining rod. So when you shine light, it have a strong series. But when we see a target DNA, microRNA, mRNA, the loop recognize the loop, open the loop, and when you open loop, you notice the dye is much further from the lining rod, so no signal or very little signal. So this is kind of a very simple bioassay that we developed to detect them. So what application? So we show first that we look at in the HIV gene, we show that when there's nothing, form a loop, you have a strong cell signal. When we have a signal like a template that have a random sequence, nothing's changed. But when you see the HIV gene there, suddenly it opened the loop and no signal. So the signals is from on to off. Later we treated also, we detect also the type of gene that found in breast cancer. So BRCA1. For example, here you see the same principle. When there's nothing, so suppose the patient go to a office, give a blood sample or something like this, and we throw in the probe. If nothing is there, the probe will still have strong signal. And here it's just a random template. But if the gene of breast cancer is there, suddenly open the probe and you have no signal. And usually I put the name of the person, like Dr. Muzendi Wabugile, who was my postdoc. He's the one who instrumental in starting this. So we want to detect not only when there's a disease, but also look at the host expression. What it means is that when a person, like for example, is infected with a disease like here, like COVID, SARS-CoV-2, then the host response. And the host response, they have some expression of certain mRNA. And Dr. Jeff Ginsburg, which is one of my collaborators at Duke, his team discovered that RS82 mRNA is the most highly expressed gene for people who are infected with viral infection, which is very important. When people get sick, you go to the doctor's office, they don't know whether it's bacterial or viral infection. But if they have RS82 in the blood, for example, they know that it is a viral infection because it really changed completely the treatment. And recently, of course, they found that people who are in ICU, they found a lot of RS82 for those that triggered infected by COVID. So we developed a probe, and as you see here is an example to show how it's worked. When you have a two probe here, and when you see this RS82, if the probe is there, look at this. When the RS82 increase, the probe decrease. When this is a control probe, nothing happened. So we can also quantify and develop this kind of a host, in fact, the host response to infection. So then, we work first a lot with a nanoparticle gold server, and then I think about 10, 15 years ago, we developed a so-called nanostar. And that, why nanostar? Because it has a so-called very sharp spike, so a strong lightning rod effect. And we, in fact, Chris Curry and Scott Tuan, two of my postdocs, they really, developed this, synthesis, and the synthesis allow them to, to control the spike, the number of spikes, and they calculate, and you show that, really we have a hotspot at the tips. And when we also increase the type of number of band, you can shift also the plasma bond to infrared. So we have also used nanostar for therapy, which is not part of this talk, but we want to have a type nanostar who is biocompatible and also can be excited in the near-infrared with the optical window to tissue. So when we do that, we develop another type of probe called Inverse Molecular Sentinel. It comes to the, why we do that? Because we work with doctors and they say, Tuan, your probe was always an on to off. Is it possible that you can do an off to on probe? So then we start with nanostar, and you see we have a probe that have a Raman label that is stretched out so it's not far from the lining rod here, and we use a so-called placeholder. So the signal is off because it's nothing there, but when we have a target will lock into the toe hole that we design here, and then it branch migration, slowly remove it, and then it removes the placeholder, and when most of it make the loop, and wait to move, it has some signal. So now we have a so-called the inverse molecular sentinel that has off to on. So now we wanted to mention that it's a homogeneous assay. That means that you don't need to do any separation. You throw in the probe, and if the target is there, you just lock it on, remove, and have a signal. So very simple. So again, Dr. Wang and Andrew Fels, my student postdoc working on this. So we try now on the microRNA mutant one, and the microRNA is a kind of biomarker of a cancer, and we work with Dr. Kathy Garman. She's a specialist on GI cancer, and what we get is to get the tissue samples from patient, and we work on really a double blind test. That means that we the number of samples will go to another group. In that group is the biostatistic group, and us we do SIRS. And as you see, without PCR, the correlation is very good, except for this, this Barrett esophagus, which is a neoplastic. All the tumors are detected by the signal of the SIRS. So we have here a type of sample based on SIRS that doesn't need to detect amplification. When we compare with PCR, the SIRS is as good or even better than PCR. So it could be a nice point of care testing as an alternative to histopathological diagnosis, especially for global health. It's very hard to have doctors, pathologists, but here you have something with objective quantitative based, it is, I think, very, very simple. So now we talked so far as in vitro test. We want to do something like in vivo implant sensors for diagnostic. So the idea is that it would be great if we have, an implant system and then have a detector that, bound to this. And, whenever the person feels sick, certainly you can see the signal. So that's the idea of the implant system. So what we did is that we work on several animal models, and I show data on this on the large animal with the pigs. So we embed the nanostar, as you see here, with the probe extended in the kind of gel. The gel is very biocompatible, and then when the target is on, it just remove and make the loop and the signal. And here we call it as a smart tattoo. So this is part of the project, in fact, funded by DARPA. And DARPA want to see, is that in the future one day, people have a tattoo and just look at the color or have a handheld reader, they can know that person is sick or not without, having to go to the doctor. So we did that. And then we work on mice, and here to show you that we work on a large model of pigs, and we were able to detect, with the signal. So we have the pig, and we inject the target. Of course, here in this case, we do synthetic target injection, and as you see, before target injection, you have a very low signal, but with the target, you see a very strong signal. And I show you Dr. Register, Dr. Wang, who really developed the system, and we work with Dr. Blitzman and Palmer. It's a big team, from different disciplines. And here we are in kind of the animal testing facility. Then from there, we want to say, is it possible for us also to do it on the chip? Indeed. So we put, instead of the nanoparticles, we coat a chip substrate with nanosphere, and we coat with a layer of gold. And then we put on top, as you see here, we put on top the nano IMS, Molecular Sentinel. And from there, we look for Dengue. Here, for example, we look at the jacket Dengue, and you see, for example, again, with a blank, no signal. When we have the Dengue sequence here, we have a strong signal on the chip. And it's very quantitative. So it shows that, yeah, from a chip platform, we can apply it for infectious disease. Then we want also to see whether what else. So before we do the molecular sentinel, here we apply the inverse molecular sentinel. So we look at RSD2 gene, as you see that mentioned before, the same thing on the chip, and we were able to have a chip. And we also not only do the nano array that we spin coat on chip, we do also micro nanolithography to have all sorts of chip substrate. And you see you have a triangular shape. You have a lot of hot spots in the middle of those are shaped, and then you have a very strong cell signal, because as you know, the more sharper the spike, the better, the stronger the lightning rod.
Tuan, we're running out of time.
Okay, very quick. So we go to another system we call a nanorattle. The nanorattle, what we do is that you see you have a core with a nanosphere, and then we coat it with another shell, and in the middle we have a dye. So in the nanorattles, it is a plasma coupling with the core and the shell that make a strong signal. As you see here on the right, extremely strong. And we design a system that use a magnetic probe, and this is a first prototype. But then you see that we can do much simpler in the next slide. And so with that, we look at malaria, and as you see with malaria, we can detect very strong signal. Look, malaria infect blood cells, and here non-infect, we can very strong signal showing that it's very effective. And lastly, we apply it to head and neck cancer. As you know, head and neck cancer is a global problem because I think, as you see from in Africa and Asia, there are a lot of risk factors due to smoking, drinking, betel nut chewing, and HIV infection, as you see. And what we want to do is something simple, because now the current diagnostic is histopathology, and by trained medical pathologists, which is missing in those low resource settings. So what we do is that we have we call a lab on a stick, which has we have different chamber, and we have a magnet here, and by pulling in and out, we just go very quick, just in 1 minute, we pull and concentrate. It's kind of very fast ELISA with magnetic, and then we test on patients. So these are the patient samples. We have 100% sensitivity and 89% sensitivity. And I mentioned that, Priya, Vora, Juan, Mo, and Pietro Strobia have working in instrumental Inverse, and our collaborator is Dr. Walter Lee. So with that, I would like to conclude that, yeah, there's a multiple plasmonic system that have proven to be very effective for detection of infectious disease, also for early detection of cancer, and also they are compatible for solution assay and chip platform. They can be also implantable as in vivo sample, and they can be simple, rapid, and cost-effective for global application. And thank you.